Hello class, so this is the first part of the series Nucleotide Biosynthesis. So in this first part of the series, we will be discussing nucleotides can be synthesized by de novo or salvage pathways, and then the pyrimidine ring is assembled from carbon dioxide, ammonia, and aspartate. So, as far as nucleotides in general, nucleotides are key molecules required for a variety of life processes. So I listed here some examples that I'll let you read on your own, but some of the highlight of things that's most well known is ATP is a universal currency of energy, and GTP is also served as an energy source for a more select group of biological processes. But there are more that are listed here, and I'll let you read that on your own. And here just a reminder about the nomenclature of bases, nucleosides, and nucleotides. So this is a reminder of what is a nucleoside. That is when you have the sugar that is bounded to the nitrogenous base. That makes the nucleoside. But once that phosphoryl group is attached, that is what makes this whole thing a nucleotide. So that's just a reminder. And as far as your bases for RNA, you have adenine, guanine, uracil, and cytosine. And then as far as the naming nomenclature for nucleoside, that is listed here. And then as far as making a nucleotide, that is listed here. And then remember the shorthand of having AMP, GMP, UMP. The M stands for how many phosphate groups that you have. So if you only have one, which is mono, that is a representation of a letter M. But if you have ADP, that means there's two, and that's pyruvate, two phosphate groups. So that's ADP, and then when you have three phosphate groups, then that is when you have ATP, where you have that T that's in the middle. And as far as DNA, just know that uracil is not part of RNA, but as far as basis for, it's not part of the basis for DNA. What is instead of uracil having that base is thymine for DNA. And just notate that when you have that deoxy, that is representing that you have a DNA, but since thymine is a base that's only in the DNA, they don't put deoxy in front of it because it's already known that this is, we're talking about DNA. Okay, and so as far as the de novo pathway, de novo pathway is actually setting up a framework for a pyrimidine base. It's assembled and then attached to a ribose or your sugar. Also, you can have the framework of purine base is synthesized piece by piece and then directly on to a ribose base. And so you have your activated ribose, in shorthand PRPP, and then having amino acids, hydrolysis with ATP, and then you have carbon dioxide, and all of that combined together to end up making your nucleotide. So remember, with de novo pathways, it's pretty much the buildup of a nucleotide. However, with salvage pathways, you can think of it like it's recycling. So you already have this base that already been made from a degradation of RNA or DNA, and then being reused and being reattached to an activated ribose, and then you have your nucleotide made again. So salvage is more so of you already have something to work with, so you're just recycling but de novo is more so building up of a new base and then attaching it to that activated ribose. So as far as the scenario of the pyrimidine ring being assembled, you use carbon dioxide, ammonia, and aspartate. Aspartate is your amino acids, as you can see that I mentioned previously here. So with that, the carbon dioxide, use it as a bicarbonate that reacts with the ammonia and reacts with ATP. And notate that there's two ATP to make this carbamoyl phosphate, which will react with the aspartate, your amino acid, to form that pyrimidine ring. And so carbon 2 and N3 atom is coming from that carbamoyl phosphate. That's why it's highlighted in red. And the rest of the carbons and nitrogen is actually coming from that amino acid aspartate. Then you attach it to the activator sugar, so the PRPP, and then you have you end up having your nucleotide that goes through some other steps to end up having UTP, and you can use UTP as a source to make CTP for the buildup of RNA, or you can use UTP to end up making TTP, where you can end up using that for DNA. The same thing for CTP. They can go through some steps to end up making DCTP for DNA. And so there are three active sites of carbamoyl phosphate synthetase. Two, that's an enzyme that is used to make this carbamoyl phosphate. And so it's linked by a channel through which intermediates pass. So this channel is highlighted in yellow here, where you have glutamine that enters and it goes through different intermediates within the channel and comes out as carbamoyl phosphate. And these channels serves two roles. Intermediates generated at one active site are captured with no loss caused by diffusion. And then as far as also with the intermediates such as the carboxyphosphate and carbamic acid are protected from hydrolysis while it is in the channel. And so you have this carbamoyl phosphate. So how do we start making this pyrimidine ring? So once again, it reacts with the amino acid aspartate. It does notate that this enzyme is aspartate transcarbamylase. And so it produces this intermediate, which gets protonated and then finally oxidized to end up forming this orotate. And so once you have this pyrimidine ring, it needs to be attached to the sugar, but remember it is the activator sugar. So how do we get the sugar to be activated to form the PRPP? So notate that PRPP stands for phosphoribosyl 1-pyrophosphate, which is listed right here. And the synthetase is the enzyme that is used to make that PRPP. And this notate to have your sugar to be activated, pretty much this hydroxyl group has a pyruvate group that is attached, so it is ready to be replaced with a base to end up making your nucleotide. Can you see your sugar? You see the phosphate group, The only thing that is missing here is the base. And so how that is happening, you have your PRPP and you have your orotate. It goes through a hydrolysis to release the pyrophosphate and attaches the base that is right here. And then from there, you have a decarboxylation that occurs, and a protonation that occurs as well, and you finally have your nucleotide that is shown here. And then from there, in order to start doing some interconversions from having that UTP to CTP, nucleotide mono, di, and triphosphates are interconvertible, where you can have UMP, which is pretty much this structure right here. We only have one phosphate group. You can utilize ATP as a source of more phosphate groups to be added to the nucleotide to make it, instead of UMP, UDP. So you have two phosphate groups that is represented. And then from there, you can add some more phosphate group into your nucleotide to end up making a triphosphate group. And so when you have this X and Y, this represents different types of ribonucleosides that are available. And so now that you have UTP, how does it convert over to CTP for RNA? So you use glutamide, and it goes through hydrolysis, and then you have your free ammonia that is available, and that attaches to that pyridine base, as you can see right here, and then you have some ATP cleavage that's happening, where a phosphate group is detached from the ATP, where you have ADP as your side product. So in order to have this UTP to convert over to CTP, you need ATP and you need ammonia. And in order to get this ammonia, you can use that glutamide as a resource from having hydrolysis that happen. And then finally, as far as the salvage pathway type of method to recreate nucleotide, so that's this recycling pyrimidine bases. So right here you have your thymine that was released from a degraded DNA, and then from there you have your activated sugar once again, and then the base can reattach to the sugar to make your nucleotide. And this particular enzyme for the thymine being reattached to make a nucleotide through a salvage pathway is listed right here. The thymidine phosphorylase. That is the enzyme name for that. And then from there, you can convert the nucleotide thymidine into convert it into a nucleotide by thymidine kinase. And that is when you end up having your TMP. So right here, excuse me, this is your nucleoside, and then here you have your nucleotide, because now it has the phosphate group. Here it didn't have the phosphate group that was listed here, because you only have right here is just the sugar and the base. But once you have the phosphorus group attached to it, that's when you have your nucleotide. And that is the end of part 1 of this series.